then went to Germany, where he did his postdoctoral training in Max Planck Institute in geometry in groups of granular in physics and in cell biology in group of In 2011, he has started his own group in Princeton in the Department of Chemical and Biological Engineering, where he studies quantum phenomena in living systems. And today he's going to teach us about one of those quantum phenomena, which will be how the cell takes advantage of phase transition in cell nucleus.
Okay, great. Thank you, Alexandra. Thanks a lot for having me. It's my first time at NYU, and it's really a wonderful place. And of course, I'm familiar with the soft matter community and the strengths there, and I've heard wonderful things about everybody else too. So it's nice to get to see you all. So I'll tell you a little bit about some of the work we're doing in my group. We're really interested in biological problems and trying to use approaches from materials physics and soft matter physics to try to understand biological systems. We're interested in this question of self-assembly, and in particular how self-assembly is able, in biological systems, is able to sort of span these multiple length scales. You know, if you think about the building blocks of life are these nanometer-sized protein and RNA, for example, and their interactions on this nanometer length scale somehow encode for information on larger length scales. So something about how these structures, organelles within cells, have certain composition, size, shape, maybe material properties, and those properties of the structures inside the cell are then encoding information on even larger length scales about whether a cell is going to grow or divide or differentiate and things like that. So we're interested in trying to distill kind of mechanistic biophysical principles that govern the connections between these length scales of self-assembly. And I'll tell you about some of the insights we've had. This is this is a very deep problem. I think many of you in the audience are also interested in this kind of a problem, and there's a rich tradition in physics of thinking about emergence on different length scales. And so we'll try to borrow from that insight, let's say. So a lot of the focus, what I'm going to talk about today is kind of at this intermediate, this mesoscale, where a lot of the action is, and sort of the bridge, if you like, between these super small and larger length scales. And this is a difficult length scale to operate at because there's not so many tools to try to probe here, and we'll tell you about some of that. Okay, so that intermediate length scale at the level of individual structures within cells. If I say the word organelle, the thing that comes to mind are these membrane-bound bags within cells that compartmentalize the interior of the cell. But there's a whole class of organelles. In fact, I think it may be fair to say that most organelles in the cell, most of these large structures inside the cell, are not membrane-bound, but rather they're dynamic assemblies. Typically, they comprise of both RNA and protein, and they go by different names. Typically, they're called RNA granules or RNA protein bodies or RNP bodies, and I may use some of these different terms, but I mean the same thing. And so these are a type of physiological assemblies. These molecules get together into these kind of dynamic aggregates, and this is important for physiology. So some of the examples, I'll tell you a little bit about these germline P granules. So this is a C.elegans embryo. It's a worm. It's a very popular model system. This is about 50 microns across, and you can see that these structures end up on one side of the embryo. And once this embryo divides, only one of the two cells, the one that's kind of in the right side in the picture here, will get those structures. And that's one of the first steps in specifying the fate of that cell. So these are RNA protein assemblies. If you look more or less in any of our cells, you'll see these processing bodies, these green puncta here, which are involved in mRNA turnover and decay. So again, somehow involved in RNA-related processes. Inside the nucleus, there are dozens of different types of these structures. Here I'm pointing to nucleoli, which is a big focus of what we're interested in my lab. And these are sitting at sites where there's ribosomal RNA is actively being transcribed. So many dozens of different types of these structures, just three examples, and somehow they're involved in, if you like, regulating the flow of genetic information, DNA to RNA to protein. They're helping facilitate that flow of information in transcription and translation, and by functioning as a type of dynamic organelle inside the cell. Yeah, Jess?
Are they encased in?
No, they're really, I mean, we have seen examples where there are proteins that maybe act like surfactants, but there's really there's no phospholipid barrier, so they're kind of open structures. Yeah, and in fact, I'll show you some examples of this, but if you do photobleaching, for example, you know about this, maybe other people don't, fluorescence recovery after photobleaching. If you say how dynamic are the molecules, they're super dynamic. They're kind of constantly exchanging with the surrounding cytoplasm or nucleoplasm. So there's really no boundary, well-defined boundary in that sense. If you ask about the concentration, there's certainly sort of a discontinuity in concentration. So there's an effective boundary, but there's no wall, there's no enclosing membrane or other structure. Yeah, good question. Yeah. Okay, so we're interested in trying to understand these structures. There really has not been, in a way that would be satisfying to an audience like this, there's not really been any kind of mechanistic biophysical understanding of the assembly of these structures, even though there's dozens of different types of them, and they're involved in a whole host of biological processes. The other reason why you should care about these structures is because they are related, and it's increasingly apparent that they're closely related to pathological aggregates inside cells. I described them as a type of dynamic aggregate that's important for physiology. We know that proteins can also aggregate and cause disease. ALS, Lou Gehrig's disease, or Stephen Hawking has this disease. He's actually, many of you know this is, he's a rare case that survived many years, but it's usually within a couple of years it's all over, and it can onset in someone's 30s. Alzheimer's, something like 1 in 9 Americans over 65. Your brain is going to do this, and you're going to have an incredible cognitive dysfunction. And again, this is a type of protein aggregation disease, and then there are the prion diseases. So we should care about these assemblies because they're important for physiology, and they're also implicated in some of the pathology. And we should care about them because, I said this to the students in the discussion. I mean, you look around, right? A bunch of us are going to get these diseases, so we should really care.
But I thought this was completely different. I thought these diseases were based on genetic mutations and misfolding.
Yeah.
The other one is all about RNA assemblies. Or are you saying just any type of?
Yeah.
Are you relating them just like saying this is
Yeah. I'll give you a little bit more concrete examples later, but just for now, I'd say that many of the proteins that are have kind of sticky interactions that promote the assembly of these physiological structures. Those same proteins, if a single point mutation, so certainly there can be genetic defects, a single point mutation, and that protein is just a little bit more sticky, and then you end up with ALS, for example. So in that sense, I mean that there's this link. And so the physiological structures might actually be kind of promoting this pathological aggregation. And some of it is genetic, as you say, although the connection between, sort of nature and nurture, the genotypic consequence versus what's happening in the environment, or, there's certainly age-related, so something happens in time. But increasingly, the connections are becoming more clear that between these things and the physiological things. And so I'll explain that a little bit more. Yeah. Okay, so I'm going to try to, this is kind of a sort of outline. I'll give you an introduction about the way we're thinking about these structures. I'll tell you something about the molecules involved in the assembly of these kinds of structures as more and more insights we have into that. I'll tell you a little bit about this, the role of RNA transcription and some modeling we've done in droplet coarsening using kind of borrowed from the materials physics approaches that are taken, and then I'll try to give you kind of a conclusions outlook on these things. And so first, an introduction to these droplets. So I've already, sort of given you the high-level introduction. I showed you a picture of these germline P granules, which are localized to the posterior of the C. elegans embryo. This is why I first got interested in this problem when I was a postdoc in Germany. Worked in Tony Hyman's lab, and we're asking very simple questions about how, what are the rules that govern the localization of these structures. They go from, these green puncta distributed everywhere, and they end up on one side of the embryo. And I won't tell you an enormous amount of detail about this. I just want to give you kind of the high-level view of what this is about. But in a series of experiments, what we showed was that these structures are not being translocated to this end of the embryo, is what we initially thought. But rather, there's a gradient in the stability of the structures across this axis, the anterior to posterior axis. And so you can sort of see this if I've labeled in blue a P granule that's sitting in the anterior, and as a function of time, this is integrated intensity, so something like its size goes down and down and down in time if it's in the anterior. If it's in the posterior, there's initially a sort of decay, but then it stabilizes and can actually grow. So there's a spatial gradient in the stability, I'll say spatiotemporal gradient in the stability of these structures that gives rise to them all being on one side versus none on the other. We started to ask, we got this data and we said, okay, this is sort of looking like maybe they're condensing and evaporating or something, and we started to ask questions, what are these structures that are forming? What is the kind of biophysical nature of these structures? And we did a very simple rheology experiment, if you like. What we did is we took the worm, there's a different place in the worm, but where these structures collect around the nuclei, and we applied shear stresses across this tissue. And hopefully you can see in this image, but when you, the dark regions here are nuclei, and around these nuclei are assembled these P granules, this P granule material, and you can see that it flows and drips. We see things that look like wetting and de-wetting, where it sort of pinches off the surface, and coalescence, and so forth. So this material on these time scales, let's say, looks like a liquid. Okay, so it looks like a kind of liquid. And this, and some other data that I'm not going to show you. This led to a model wherein these P granules in this segregation process, now it looks like these are droplets of RNA and protein that are condensing in the posterior of the embryo. And so the picture that emerged was that this anterior-posterior axis spans a phase boundary. Yeah.
Can you give me 2 seconds to mess with the lights?
Sure.
I'll probably make it worse, but
okay, sure. Yeah.
There's a panel here that's supposed to have mood lighting.
Can people see that, sort of? Not so well? Yeah, thumbs.
This is AV.
So but on the left is single-cell embryo, is that right?
Yeah, this is...
And on the right is after several cells.
Right, so this is what the embryo looks like basically right after it's fertilized, before there's any detectable asymmetries across the anterior-posterior axis. And then the embryo starts to... it breaks symmetry. Thanks, that seems helpful, yeah. The embryo breaks the symmetry and establishes basically a gradient across this anterior-posterior axis, and concomitant with that is this differential, stabilization.
So
and
it looks like it's, I think I can see the pronuclei there. On the right, so this is during migration of the pronuclei.
Right, so you're referring, this is the paternal pronucleus that's contributed by the sperm, and this is the maternal. And then here you see them where they've just met, right, so this is kind of they're starting to meet, pronuclear migration during this process, yeah. Great, okay, good. So the bottom line is it looks like this embryo is spanning a phase boundary where the posterior is in this two-phase region where droplets are condensing, and the anterior is, sort of where they're dissolved. So we started to say, okay, is this specific to these types of structures? Maybe these P granules are just happen to be a special case, or maybe is it more general? This idea that RNA and protein can form something that looks like a condensed liquid phase in the cytoplasm and can assemble by a type of phase transition. And so we did some studies in this frog system. This is a completely different system. It's basically a single huge cell. It's about 1 millimeter across. It has a single huge nucleus. It's about 400 microns across. It's bigger than most cells. And inside this huge nucleus, there are actually hundreds of these nuclear bodies, are RNA protein bodies. Here they're nucleoli. There are other types in here, but most of the ones you can see are nucleoli. And so what we showed, again, some sort of simple mechanics experiments, we showed that if you push 2 of these droplets together, they readily round up and fuse into a single larger droplet. And if you you disrupt this, after they've started to fuse, you can manage to pull them apart. You get things that look, like classic liquid bridge rupture between these two bodies. So this is now liquid-like behavior on pretty fast time scales, tens of seconds. You see this nice liquid bridge rupture. So this, and the size distribution was consistent with these sort of slow coalescence events that happen as a function of time. And so it looks like maybe this idea is more general. These structures behave as liquids, condensed liquid phases of the, in this case, the interior of the nucleus. And so, this is kind of a cartoon, for a physics audience. This is sort of maybe embarrassingly simple, but you give talks to different groups. And in any case, it's useful to say, okay, gas, liquid, solid. And, we're used to this idea of phase transitions as an incredibly powerful framework for thinking about the organization of matter and how it crosses these different length scales. And sometimes, occasionally the biologist will say, okay, but this is sort of what you expect for water or small molecules, but we're talking about, proteins and RNA. We know that proteins can undergo phase transitions. This is X-ray crystallography. You put high concentrations of protein in a test tube and it makes these crystals. And under other conditions, you can also get protein liquids in a test tube. And so essentially what we're saying is that this is happening inside of a cell too, and really, why should it not? I mean, there's high concentrations of these molecules inside of the cell, and so it looks like it's happening inside cells. So this idea has started really increasing evidence for this idea over the last few years. I mean, this is a fun story that I won't tell you about, but we've in this huge single cell with this huge nucleus, we've shown that these droplets really are kind of an emulsion of RNA protein, these RNA protein liquid phases, and we can destabilize it. And actually, gravity is important because the cell's so big, and so all these droplets rain down and fuse into a single larger one. So this is basically there's an actin network that stabilizes this emulsion. And something like salad dressing. And you can induce nuclear bodies. This is in a Drosophila system. People have seen evidence for a kind of liquid phase separation for stress granules, and there's a few different papers in this area now. This is a nice paper from Mike Rosen's group showing that these multivalent proteins can drive phase separation in vitro, and if you express them in cells, you see things like the organelles that I've been describing. And this is a really nice paper that just came out, this year, let's say, Molecular Cell, showing some really quantitative agreement with the expectations of classical phase separation. And there's a bunch of papers from other groups. Yep.
In your first example, in order to maintain this gradient across the cell, you have to do something. There should be some active process.
That's right. Yeah. Yeah, and so I've kind of glossed over that, but you're totally right. I mean, that's a clearly non-equilibrium situation. It's the analogy is like setting up a temperature gradient with droplets across the, and I could tell you a lot about that. There's essentially a morphogen gradient inside this single cell that is maintained by differential phosphorylation, which changes the diffusivity of these molecules. It's a fascinating gradient.
Phosphorylation happens at the poles.
It happens essentially at the poles. There's some evidence that it can happen inside, but you could think about it more or less at the poles, and this sets up a gradient. Yeah, it's a really neat example. Yeah.
But at least in that first example, we have the P-granule
Yep.
density over the posterior.
Yeah, that's right.
So, I mean, that's also, I guess, when you have cortical contractions, so presumably driving the female this towards the
That's right. There's probably I can.
So
yeah.
you're kind of saying you're kind of having to maintain a gradient against the cytoplasmic flow.
That's right. So, in fact, the idea in the literature that was in the literature for about 15 years was that these P granules are advected with the flow. And that's why I became interested in that problem, because you might say, oh, that's nice, they just get carried over, but it's a closed volume, and it's like a cavity flow problem, so you have to have backflow, and then you need something else, like a catching mechanism. And in fact, when you measure it, as many cross into the posterior as cross out, so it looks just like a cavity flow. But with respect to the gradients, these are low Peclet number situations, even though they can advect the granules because their diffusivity is low, Yeah, good questions. Okay, so I'll tell you some about molecules and properties now. So what's really driving these phase transitions in cells? So, the standard paradigm of proteins, we know protein is a linear polymeric chain, but the action comes from this protein folding, this protein folding problem, right? Really rich. You basically usually think about a deep free energy landscape, a deep well that folds you into this single well-defined conformation, and that's how proteins have their enzymatic activity and binding partners and so forth. But this doesn't always work out, right? Some proteins are conformationally heterogeneous. You can think about shallow free energy landscapes where you don't really go into a single well-defined conformation, but rather there's a whole host of conformations, and this is typically referred to as intrinsically disordered proteins. And there's a lot of excitement in that field right now because more and more people recognize that the whole paradigm of well-defined protein, folded proteins is only a part of the story. Lots of proteins are intrinsically disordered, and we can think about them as kind of floppy chains that interact in more complicated ways. And it turns out that these intrinsically disordered proteins, more and more it's starting to look like they could be relevant for these phase transitions. I'll tell you a little bit about that. So if you look, people have done bioinformatic analysis of proteins that are predicted to be disordered. And this is just some examples in these germline P granules that I've told you about, some of the components that have regions of predicted disorder. There's one that's not on this list that we became interested in. It's called LAF1. It stands for lethal and feminizing. And that just should imply that this is an important protein because it's involved in sex determination, and if and more importantly, you just basically die if you don't have it. And so it's co-localizes with other markers. This is a protein called PIGL1, which is a classic P granule marker. So they basically both are in these structures, and this is a prediction of the disorder. So this is kind of the N to C, N terminus to C terminus. So it looks like there's an N-terminal domain, this arginine-glycine-rich domain, which is predicted to be strongly disordered, and there's a similar disordered region on this end of the protein, whereas the middle is probably fairly well folded. So we became interested in this protein, and we have some collaborators in Dresden that we've worked with for a while, and we did, we've done some of the experiments ourselves as well. That if you do RNA interference on this protein LAF1 to basically remove it, these P granules are basically completely disrupted or very strongly disrupted. Okay, so this protein PIGL, classic P granule marker, you don't see it much. Maybe in a small number of granules remain when you remove LAF1. So it seems like an important protein, and we wanted to try to understand what's happening from a kind of bottom-up perspective, because inside the cell is very complicated, and there's a lot of proteins, a lot going on. And so we've purified this protein LAF1. This is some work by a talented postdoc in my lab, Shani Elbaum. And what she's been able to show is that LAF1 on its own at pretty physiological concentrations forms these beautiful droplets in a test tube. And so here you can see, this looks like your windshield in a rainstorm. These droplets are sinking down to the bottom and wetting the surface and then coalescing with one another. And so, I showed you, that these P granules in the living embryo have liquid-like behavior, and it looks like a phase transition, some kind of liquid-liquid phase transition in the cell. And here's a key P granule protein that in vitro alone will do this. And so we started to map, we've mapped out this phase diagram, and this is salt concentration. Many of these things really look like strong salt dependence, so electrostatics is important. But if you say physiological, maybe 150 millimolar concentrations, and then the phase boundary is at 1 or 2 micromolar, which is near physiological protein concentrations. So if you're in this region, lower salt and higher protein, you're going to get these beautiful droplets. This is an example of droplets that were wetting on the surface, and then we've dumped in high concentrations of salt, and you can see them basically dissolving, all dissolving away. It's kind of beautiful the way it works. The way that it happens, but I can tell you more about that later. And so we've been. I won't. I don't think there's too many structural biologists in the audience. I won't go into too much detail. Suffice to say, what we've been able to show is that this disordered arginine-glycine region is necessary, and it's necessary you get rid of it and there's no droplets that form in vitro, and it's actually sufficient, so this domain alone undergoes this liquid-liquid phase separation in vitro. And so that's this disordered domain. We've done some circular dichromosome measurements that have, to prove that it is a disordered domain. If you're interested in protein structure, you may want to sequence gaze. I mean, I guess most people are starting to glaze over looking at this in this audience, but it's interesting because it's very common. You'll see basically stretches of glycines and some arginines, these positively charged arginines, and you'll get aromatic groups like tyrosines that are kind of sprinkled in. So there's a sort of, let's say, quasi-repetitive nature of this. There's kind of blocks of domains, which might be important. Yeah.
So when you say liquid-liquid phase separation,
Yeah.
you mean between pure water and water rich in these proteins? Or
Right.
do you mean some other buffer?
Right. What I mean by liquid-liquid phase separation is we're thinking about, one liquid, which is, you could also say, if you like, I mean, this is the dilute gas-like phase of the protein molecules but
Like that. I don't know how to think about
yeah. Do you?
gases.
That's right. Well, that's why I call it a liquid, right? Liquid-liquid phase separation. So it's one liquid, which is the dilute protein in buffer, and then there's two liquids when you cross into this region of the phase diagram, low salt and high protein, and that is high concentrations of protein in these liquid droplets coexisting with a dilute protein solution that's, buffer and protein.
Oh,
Yeah.
so diluted solution of proteins and plus. So rich in that protein.
That's right, or poor. That's right. Yeah, and the enrichment is like factors of 10 to the 2 or something. It's much higher concentration in the droplets. Yeah. Good. So, okay. So we wanted to try to, I've been calling these things liquids, right? And we wanted to try to understand, are they really liquids? I think I used the term, low budget rheology experiments. These are still fairly low budget, but maybe slightly higher budget, I guess. We tried to use microrheology. Okay, so we did very simple passive particle microrheology. This is in vitro. It's in equilibrium, and so we've introduced probe particles, micron-sized, pegylated beads into these droplets, and then asked what is the mean square displacement as a function of lag time. So fairly simple experiments, and so the mean square displacement looks to be quite linear with lag time on these time scales. And so then we can interpret this from the Stokes-Einstein relation and try to extract out the viscosity. And so what you find is that the viscosity of these droplets, so they look purely viscous on these time scales, and the viscosity is something like that of honey. What's interesting about this is that RNA, so those experiments, there's no RNA. If we add RNA to this system, because RNA is present in these droplets inside the cell, if we add RNA, then the mean square displacement increases, which, of course, that corresponds to a decrease in the viscosity. So RNA seems to effectively, we could say, fluidize, if you like, or decrease the viscosity of these droplets. You can see that. This is from the mean square displacement from the microrheology. You can also see this from the molecular dynamics within the droplet. So we've done these photobleaching experiments that I mentioned briefly before. This is you have fluorescently labeled proteins inside the droplet, so all these proteins are labeled, and then you basically bleach the ones in this little region in the middle and ask how long till that signal comes back, because the bleached guys are exchanging with the unbleached guys. And it's basically the same thing that you might have expected from the microrheology, that when you add RNA, the signal comes back much more quickly, and so the viscosity is decreased, the molecular dynamics within the droplet is increased. So this is kind of interesting because it gives us some idea about what sort of material we're dealing with, and how RNA may change the properties and molecular transport within the droplets if we're thinking about them as little liquid-like microreactors within the cell. Okay?
Do I understand correctly that this protein is predominantly positively charged, but he has also some negative groups, so it's polyamphylite?
Yeah, that's a good question. I mean, these are polyinfolitic. I mean, there's positive and negative charges. LAF1, the protein we're focused on here, has a slight net positive charge.
Which is why
These arginines.
is with RNA.
Yeah, although there are other proteins that seem to have. So this is not really clear. We've worked with some, a few different ones, and so some of them seem to have more blocky. There's this nice paper that I mentioned by the first authors, Julie Foreman K from 2015 moleculars. They have this protein which is actually very similar to LAF1, where but it's more blocky. The charge distribution, it tends to be positive charges and then negative charges. So it looks a little bit like, like a block of polymer of some kind. And they showed that if they shuffle those charges, then it doesn't phase separate very well anymore. LAF1 doesn't have such a well-defined blocky character. But yeah, we're trying to understand what that, and dig into the literature of complex coacervation, for example, or regular coacervation to figure out what that all means. I think it's nice because I think new physics will emerge because you're in the realm where conventional polymer physics ideas can apply, but you're sort of pushing the limits because you get into the whole sequence space issue. But I think it's more I was mentioning to the students, it's probably a bit more tractable using those ideas than, for example, the protein folding problem. So, good questions. Okay, so I want to mention. So I say, what about the in vitro gels formed by Purified? So I say this because there was a series of papers that came out a few years ago that have been really influential, where, showing that many of the proteins, some of the proteins that are in some of these granules, let's say it the right way, can form gels in vitro. And so a lot of people have been saying, these granules are in cells are gels, and they're formed from a kind of fiber scaffold. And that's completely different than everything I've been saying, right? I've been calling them liquids, and I showed this liquid behavior and viscosity. And so, sort of what's, the deal with that? And so we've been thinking a lot about this and trying to understand. I mean, we noted that, in these experiments, the proteins are at really high concentration, and they tend, these experiments are done typically leaving the proteins in the fridge for a while, and so wasn't clear if if these gels would be physiological. And so we kind of proposed this picture that what's happening is in cells, you've got, if you like, I think Jasmine said she didn't like this, but maybe like gas, liquid, solid, if you like, or a dilute protein phase. You can condense into these liquid states, which we think are physiological, dynamic, liquid-like organelles, but then you could transition into more solid-like states. And so this may be this part of this connection between the physiological organelles, these liquids, and the pathological things. And so just very recently, there's evidence that this is what's happening. So this is a really nice paper that just came out from Tony Hyman's lab, where they basically used exactly those same proteins that form gels in these previous papers, and showed that they, at more physiological conditions, they form liquids. Now, if you leave these liquids over time, they'll kind of evolve and coarsen. And sometimes so this is WT is wild type. So over time, you'll start to see some fibers in some of these droplets. And so that's interesting. So maybe there's a transition that can occur with these natural proteins. This is a point mutation, this G156E, and this was chosen because it's one of the mutations that's found in ALS patients, this Lou Gehrig's disease. And what they find in vitro is that you still form liquids, but they age more quickly into these aggregate states, so they gel and form these crazy fibers. And so we've seen something very similar. This is a paper that just got accepted with a poly-Q protein. These poly-Q proteins are, like for example, Huntington's. It's an aggregation-prone protein. And again, it forms droplets that can mature into fibers. So you basically can nucleate fibers within that may be pathological. So that's what I'm going to say about that. And I want to, in the time remaining, I want to tell you a little bit about this RNA transcription and droplet coarsening. So we'll see. I may have to go a little bit more quickly. But I mentioned, right, that if you can think about these bodies as being involved in regulating the flow of genetic information, DNA being transcribed into RNA and translated into protein. The nucleolus that I showed you these movies of liquid-like behavior in the frog system is strongly implicated in this. And in fact, these liquid-like bodies assemble around sites where ribosomal RNA is being transcribed. So there's the DNA, it's spitting off RNA, and at those sites, nucleoli are sitting, and probably what they're doing is creating a microcompartment where all the reactions that have to take place to process that RNA are happening right inside this droplet. So we wanted to think about, okay, how are the assembly and properties and function, right? I mean, how is this all, this liquid phase separation picture, how does this all work in regulating the flow of genetic information? And so this is this is a C.elegans embryo. This was just like the pea granules I showed you at earlier stage in the embryo. So there's a 50 microns across the C.elegans embryo, a bunch of different cells at this stage. You can see now labeled in green, this is the nucleus. You can see it kind of in this light green. And these darker spots, these are the nucleoli, so the two transcriptional sites, presumably, that these structures assemble around. So this is some nice work by a former postdoc in my lab, Steph Weber. And so we took advantage of the C.elegans system because we had a really unique handle. We were able to make the embryos bigger or smaller using RNA interference. RNA interference is actually discovered in C.elegans, and it's very easy to use. And so you don't have to worry about the details. Just we have these different genetic perturbations that we can make the embryo either smaller than the normal case. So the whole embryo is smaller, and so at the same stage, right, all the cells are smaller. We can also make the embryo bigger. And we wanted to ask this question, trying to play with concentration and move around the phase diagram to test this idea that nucleoli assemble by a phase transition. And so I'll ask for audience participation here. Do you think that the nucleoli get bigger or smaller when I make the embryos bigger and smaller? What do you think? So when I make the embryo bigger, do you think nucleoli get bigger or smaller?
Of course, smaller. Otherwise, you wouldn't. Right.
Of course, I wouldn't. You see, that's why physicists are. I don't. No, seriously, this is good. Yes, of course, smaller, or else I wouldn't be telling you. And that's the right answer.
Are telling us. They're getting bigger as you go down.
Sorry.
They are.
That's right. So this is showing you that I can make the embryos bigger, and then the question is the structures inside. Do they get bigger or smaller?
Bigger. Is that one embryo?
These are the cells. The cells definitely get bigger at the same stage. Yeah.
The embryo is a
4-cell stage. Yeah. I'm showing you 4-cell stage here.
asking about the drops.
Yeah, the drops inside.
What the cell was and what the embryo was.
Yeah, no, that's okay. So the drops inside, and you're right, something interesting happens, or else I would make a big deal about it. But we were genuinely surprised by this, right? We thought they were going to get bigger. In fact, they get smaller. So this is what the normal thing looks like. We make the embryo smaller, and these organelles get bigger. We make the embryo bigger, and the organelles get smaller.
I find that surprising.
Good. Thank you. Well, we did too. So much so that we, did one of these for months, and we thought there was no place for, physics and modeling and stuff like that in biology. So it turns out we think we can explain this in a very simple way, and I'm going to sketch it out for you because I want to get to something actually more rich. But we think that the reason for this is that this embryo is loaded. We've shown this. It's loaded with a set number of molecules. And if you load the embryo with a set number of molecules and you make the embryo bigger, then, if concentration equals number over volume, then the concentration has to go down. And so as you do this, the concentration is going down. Now, that's a nice explanation, but you'll recognize that what you need for that, the key ingredient in order for that to work, is that you would have to have a kind of equilibrium with a dilute phase. So if you took all that material and built a structure out of it, then the size wouldn't change at all. But if you have this kind of a phase picture where there's the condensed phase is in equilibrium with a dilute phase, which is at some saturation concentration, then this will happen. If you make the embryo bigger, the concentration goes down, and eventually you'll basically hit you'll hit the phase boundary and nothing would assemble at all, which is sort of what you're approaching here.
Is what you just said, and I'm confused about what you just said.
Okay, yeah.
Is what you just said that in reducing the cell size,
Yeah.
you're just pulling water out of the cytoplasm? Is that something rather than the species that are...
Yeah, the way, so there's things that I'm not telling you to keep it simple, but the way this works is that the worm is basically kind of like pinching off bags of cytoplasm that defines the embryo, the oocyte. And so it's pinching off these bags. So by and large, that bag of stuff is it's the same concentration, you're just kind of pinching it off. But this structure, the nucleus and these nucleoli, those are loaded as intact units. So it's not so in that case the concentration is not fixed, the number of molecules is fixed, and then the concentration varies if we basically change how the worm is pinching off the bags, if it's making big bags or small bags. That's basically what it is. Yeah.
So can you plot the graph?
Yeah, so I'm gonna, right, so that's exactly right. What did I show you here? This is a shame. I can show you more. I'm sort of glossing over this because I'm trying to get... But basically, we built a very simple model for this, and it works really well. It's a toy model in the sense that it just says what matters is the degree of supersaturation. So the concentration in the nucleus above this saturation concentration. So this is kind of a toy model, and it basically gives you this qualitative, in fact... It goes right through the data actually quite nicely, but it's a toy model and it doesn't explain everything as I'm going to tell you. I can tell you more about how well it does with this. We've measured the precise sizes and all this stuff. But using that toy model, we were able to put together what is a preliminary pseudo. This is kind of we'll call this a preliminary phase diagram. Okay, so. The idea is that the deeper into this two-phase region, the higher the concentration, the bigger these structures get. That's kind of what you expect, right, in a simple phase diagram picture. And as you approach this phase boundary, the things get smaller and smaller. And in fact, there are blastomeres that we were able to play with very early stages where they actually normally don't assemble, and we could induce them to assemble by increasing the concentration. Now, where I drew this on the y-axis is really arbitrary. Okay, the y-axis here is supposed to represent a kind of, this is chi, like a Flory chi parameter. So we think that the embryo is somehow regulating the strength of the interactions developmentally, and so we can actually, we can access places where there's no nucleoli detected and we cross in. This is kind of a high level. I've skipped some stuff because I want to tell you the real model that we've moved to. Yep.
Is that black line real?
No. This black line is made up. Yeah. This black.
For example, if a point is lying on the black line, does it mean that like 50%?
Yeah. Yeah. Okay. So that's good. That's why it's made up, but it's made up in an intelligent way. Let's say. So this RNAi condition is in fact very close to the phase boundary. So some of these embryos, you'll see little nucleoli, and some of them you don't. So it's really right there. Yeah. So I think that's your question, and that's exactly right. Yeah. Yeah. And of course we go the other way, make the embryos big, decrease concentration. We never see anything. So we think this is a phase boundary. We don't really have any idea what this means, but yeah. Okay. So, and the reason why this is kind of this is a toy model and it doesn't capture everything and leaves you left a little bit unsatisfied is that it doesn't capture any of the dynamics. So if I actually play a movie of this process, this is an embryo that's going through a few cycles of cell division. The nucleus is forming, the concentration is going up as these nucleoli, the components are pumped into the nucleus. And you can see that, in fact, initially you get droplets that are nucleating everywhere inside this nucleus. They mature around only two of the sites, which are presumably those transcriptional sites. So the data I showed you before was kind of the end state of this system. But there's a whole bunch of dynamics that are very rich, and we want to try to develop a better model that sort of takes this into account. I see you playing with the clock up there. Does it still work? I've been paying attention to it. Hopefully it still works. Okay, so we played a similar game to what I told you before. We have this complicated embryo system, and there's a lot going on, and we don't have full control over all the knobs because it's alive and so forth. And so what we tried to do here was to take an approach learning from a simpler in vitro system to gain some insight. Okay, and I'll try to bring it full circle, and I'll come back to the in vivo very shortly. But these are two proteins that are highly conserved, that are localized within these structures. They have these arginine glycine-rich regions, predicted disorder regions, that if you squint, they look very much like that LAF1 protein, which I showed that domain was necessary and sufficient for forming liquids. And sure enough, in vitro, these proteins form beautiful liquids, at close to physiological concentrations. I'll show you the phase diagram in a second. This is this GAR1, these beautiful droplets. And so we wanted to try to look at this system and compare what happens with the in vivo system. Okay, so if you're at a high salt concentration, these droplets don't, you don't see droplets. If you quench to a low salt condition, you nucleate droplets, and if you wait over time, these droplets coarsen. So sort of in some qualitative way, this looks sort of like what's happening in the cell. So far, this is, potentially a useful framework. So many of you might know these kinds of coarsening processes. You get often you get power law growth in things like the average radius of these droplets. And the one that I had known about before, and probably you had seen before, is the 1/3 power growth law, right? Average radius of droplets, this Lifshitz-Syazov growth law goes like t to the 1/3, like time to the 1/3. Just a word of caution, though, you can also get this t to the 1/3 growth law through Brownian motion coalescence in the absence of Oswald ripening. But we would expect something like this for actually both regimes, if there's Oswald ripening or if there's no Oswald ripening and there's Brownian motion coalescence. And in fact, we've measured the dynamics of this in vitro system when we quench into the two-phase region, and we see mean droplet radius as a function of time has this 1/3 power. People freak out about decades and all that. But I think this is pretty convincing to me that you're seeing 1/3 growth laws. So you'll notice that I've got plus and minus RNA. When I add RNA to this system, I still get the 1/3 growth law, but apparently this coefficient is changed, right? So it looks like RNA, the growth is faster in the presence of RNA, so RNA is somehow promoting this coarsening of the droplets. Okay, and we can try to estimate what, based on some other measurements, what this prefactor might be. And it looks like it's most consistent with the Brownian motion coalescence regime rather than the Oswald ripening regime, but we can't completely distinguish those. So this is what the phase diagram looks like. Again, all in vitro. This is salt concentration. This is one of these proteins, Fib1. It's got this phase boundary, physiological, concentrations. You get micromolar protein. The phase boundary is at micromolar protein, which is similar to what we have measured in cells. Now, I want to draw your attention, though, this again, if I add RNA, the phase boundary is shifted. So what that means is that if I was at a point here, right, if there's no RNA, then I would not have any droplets. I'd be outside of this two-phase region. But once I add RNA, now I'm inside the two-phase region. And so that, of course, means that RNA is promoting droplet formation. It's somehow promoting condensation of these droplets. The students are kind of very bored because I told them all this before. I told you guys you didn't have to come. We gave the... Okay, so RNA seems to promote condensation. So does that give us any insight into what's happening in vivo? Remember, all this is in vitro, so purified systems to try to understand. And, you know, it makes a lot of sense actually because this data, because RNA is being made inside these nucleoli, right? The two spots that you end up with are presumably that's where the ribosomal RNA is being transcribed. And so if you think about this kind of a movie of the dynamics, you're initially nucleating droplets everywhere, but they're resolving to just two spots. And those two spots are, in fact, that's where the ribosomal RNA is being made. So if you use oligo fish probes, the RNA is sitting at these two big puncta that are going to mature, and the puncta that don't have RNA are going to dissolve.
What are all the little spots before you end up with two spots?
Yeah. So I think the picture here is that this protein, you know, thinking back to this in vitro experiment, right? This protein on its own, and I think this is true of many of the proteins we've looked at that comprise these droplets. It can phase separate on its own under physiological conditions. So if you just increase the concentration, it's going to make droplets. But if there's RNA around, it prefers to be. You can see that because of this phase boundary is expanded. The region over which you have, you know, two-phase coexistence is larger now. So the system prefers thermodynamically to have RNA around. And so at this point, I'm just sort of hand-wavingly saying that what seems to be going on is that when you start to transcribe at those two spots, those droplets win out thermodynamically. And I'm going to show you a little model that sort of captures that. But it sort of makes sense based on this picture. Okay, so it also makes sense if we can use RNA interference to turn off transcription. If we turn off transcription, we get these little spots that basically do the same thing. They nucleate, and there's some coarsening of them, but they never, you don't end up with just two spots at those transcriptional sites because nothing's being transcribed under these conditions. Eventually, everything goes away. This is, we think the nucleus actually grows so big that the concentration goes down, and eventually the nuclear envelope breaks up. So droplets can condense on their own in the absence of transcription, but transcription, drives the preferential. So, I could go through this fairly quick. I mean, this is the kind of simple picture. This was motivated by, this imaginary, and by imaginary I mean pretend or made up chi parameter right on my axis. This is like a Flory chi parameter. This is the simplest thing, you'd write down a type of Flory-Huggins, and I don't even have the chain length here. It's n equals 1. This is like the free energy as a function of concentration. Here, phi is like a concentration or volume fraction variable, and chi is this Flory chi parameter which governs interactions. These first two terms, this is about entropy. This is the entropic contribution. The system always wants to be well mixed. If you just listen to entropy, that's what those two terms do. And this term, depending on the size, the magnitude of this chi parameter, this term can prefer the system to be phase separated. And that's the Flory chi parameter. So the energetic cost of having polymer next to solvent compared to the homotypic interaction. So if this chi parameter is really large, polymer does not like to be solvent, and you would rather phase separate. Right? So larger chi means larger driving force for phase separation. And so you can see that, this kind of schematic. This is basically from here, chi equals 0, that's just entropy. And then larger chi, you start to get this double weld shape, and you drive phase separation. This is just a review that we wrote that goes over some of that. Okay, so that tells you whether or not you're going to phase separate or not. But the dynamics to get there, you need some kind of dynamical equation. And what's typically done is, in materials physics, is these kinds of Cahn-Hilliard models. You can recognize this as a type of diffusion equation. This gamma is a diffusion coefficient, and this could just be, it would be the diffusion coefficient if this were just phi. This is an effective chemical potential that's defined by the derivative of the free energy as a function of concentration, and this is the so-called square gradient free energy term. So if you put in the Flory-Huggins free energy that I just wrote down, and take the derivative and put it in, you get something like this. And so we basically I have a couple nice theory collaborators, Nico Hatay and Joel Berry, and we tried to model this. And the picture, it's very simple. What we did is we just said, locally, we're going to define an elevated chi, which means an elevated increased thermodynamic propensity for phase separation locally at those spots. And we're going to watch the evolution of the system for parameters, reasonable parameters as far as we can measure. And this is the kind of thing you get. Initially we nucleate droplets everywhere, and then we basically elevate the chi parameter locally, which is modeling ribosomal RNA being transcribed there. And then, sure enough, these guys win out. So it's like an Oswald ripening type of effect, right, where you've got a local preference for this thermodynamic preference due to the RNA being transcribed there.
But who decides where this happens? Is it somehow self-consistent, or?
You mean in the biological system?
No, in your model.
In the model, we just define a spot that is modeling the transcriptional site where RNA is being localized.
You point by finger.
Yeah, we point and say chi is larger there and evolve the system, basically. Yeah. So, okay, so this is a little bit, at least you'll grant, this is a bit more realistic than the toy model that I told you, because it gets at some of the dynamics. And so this is what these simulations look like for different parameter regimes. You actually get a half power growth in the beginning, which we never can access. This is very early. And then the 1/3 growth law. And then once the nucleolus takes off, this basically transcriptional activation, you take off there. And this looks very similar to what we see in the in vivo system. I am not going to claim that this is a 1/3 power law. I'm aware of people who freak out about power laws, and certainly this doesn't look. You wouldn't bet any money on this, but it's consistent with the data that I've shown based on in vitro, the simulation, and so forth. And so we think this picture sort of makes sense. One of the I'm going to finish I think we started a couple minutes late, so I'll finish. Just a minute or two. One of the key predictions of this picture is that if you could adjust the concentration to get into that region between the two phase boundaries, the plus RNA and the minus RNA phase boundary, you could get to a place where you only nucleate droplets at the transcriptional sites, but nowhere else. And we can play this game by changing the size of the embryo to get into that spot, and in fact we can access that where these concentrations you basically get nothing formed ever. These concentrations you get nucleoli at those transcriptional sites, so that's kind of like in the spot between these phase boundaries. And then at higher concentrations we get droplets everywhere, right? We get these ones nucleating, and then at the transcriptional site. And there's some more predictions of this model which seem to be borne out. So this is the total amount of droplets that nucleated as a function of concentration has this this concentration dependence, and it's consistent with the prefactor fit prefactors that we can measure and from other experiments. And then the density of these droplets, again, this is like this experiment that the density of these droplets as a function of concentration. So I'll just tell you now. I'm just going to wrap up the last slide or so. I think I'll jump to this. We like this because the nucleolus, because it makes ribosomes, which is the machine that makes the hardware of the cell, it's implicated in driving cell growth. And what I've just been telling you suggests that the concentration dependence of nucleolar assembly links its assembly and function to cell size through this phase transition. So that's something we're very excited about. And then just to give you kind of a high level overview of what we're thinking about, and what I see as something we're going to be occupied with for quite a while is this picture. How these phase transitions impact the flow of genetic information more generally. I mean, you can think about the liquid, what I'm calling physiological liquids inside cells that promote this flow, DNA to RNA to protein, and the pathological solids, which, this is like, I think, Niagara Falls frozen. So basically, if you kind of lock things down, then you're going to inhibit this flow of genetic information. And we're testing a lot of those ideas. I can tell you more. So I told you this was our big picture. I'll give you 3 take-home messages if you forget everything else. RNA protein bodies represent liquid droplet phases of RNA and protein. I think the evidence, let's call this still a hypothesis, but the evidence is really mounting that some version of this statement is true. Okay. And I told you a little bit about this protein, this LAF1 intrinsically disordered protein, as one of many in this class that seems to drive phase separation. And then I told you that RNA, it can play an important role in the properties of droplets. I told you a little bit about that with P granules in our microbiology data. And then also the assembly and shifting this phase boundary and promoting condensation at a certain transcriptional site. So RNA is really playing an important role. And that's it. So I think I mentioned people as I went, and thank funding and my collaborators that I mentioned as well. And thanks again for having me. Appreciate it.
So I was wondering, you probably want to know what the driving forces are for the droplet interaction or for
That's right. Yeah. Yeah.
You didn't show any data on what happens for example, their temperature. Would that be one of the things that would distinguish between whether this is polymer physics driven or protein-protein interaction driven, so that things work only at 37 degrees?
Yeah. We have not played with temperature that much. I mean, there's some work people have seen, both lower critical solution temperatures and upper critical solution temperatures, and getting some insight into what motifs are relevant, depending on the temperature. We have not done a lot of that. Electrostatics has been our focus. Yeah.
Is it about in your free energy?
Yeah.
The driving force for the segregation is what?
You mean at the level of individual amino acids? Well, okay, so it's not. So the short answer is really it's not clear at this point. But some of it has to do with electrostatic interactions driven by, for example, these arginines. But these aromatic motifs, some of really strongly hydrophobic, like phenylalanine. And you would say in the context of protein folding, you'd say, okay, these are the guys that you want on the inside, away from the water, and they're driving the folding. Probably what it is there's dynamic charged motifs, disordered motifs in stretches of, let's say, 5 to 10 that are separated by aromatic motifs. And those guys sort of want to collapse, but the other guys don't. And by that specifically, you get pi stacking and dipole, these pi kind of things. That's happening dynamically in a way that you don't get strong collapse. And so I think that's probably why we see these kind of charges and glycines, and then they're sprinkled in aromatics. Now this is very similar to what people see in nuclear pore proteins, these FG repeats. It's glycines, phenylalanine, and there's a lot of really interesting phase behavior of those guys that is probably very similar to what we're seeing. So, but that's kind of the state of it right now. Nobody really knows exactly, exactly. There's a lot of hints, but exactly how it works is not clear right now. Yeah.
Could you comment more on the function of the droplets?
Yeah. Right. This is a key question. At this point, it's not totally clear what this is. I mean, it looks like, though, and I think the safe answer is that just like organelles that were taught in high school about membrane-bound vesicle-like things, and they compartmentalize the cytoplasm, they increase certain reactants at certain places to speed things up or store components. It's probably functionally identical what these guys are doing. They just can do it in the absence of a membrane. Maybe in some ways they're doing it even smarter, right? Because they're only assembling sort of like the nucleolus. It's only assembling when it has sort of substrate. When RNA is there, then you condense these things preferentially.
There's no specificity on the RNA.
There's also, this is also not clear. I mean, the stuff that I showed you with the phase diagram shifting, that looks not particularly specific, actually. We can put a bunch of different RNAs, so it may just be that the concentration of RNA is so high at those transcriptional sites. But there's this paper that's, I mentioned, showed you a panel of it, just accepted from Molecular Cell. There's some of this phase behavior is very strongly RNA, the specific substrates of proteins. Whether or not you phase separate depends on the type of RNA. So there can be specificity. In the example that I showed with the nucleolus, we actually don't see, there's not a huge amount of specificity in how RNA is driving phase separation, but there are examples where it is. Yeah.
Then you look at your experiment where you're in the low droplet.
Yeah.
Yeah. Do you still get your protein to the same yield?
Sorry, what do you mean? What are you thinking?
Well, if you just have your soup where you don't have the liquid phase separation, but you still have all your ingredients, are you still able to are they still able to produce proteins? And can you?
Oh, you mean? I think I know what you mean. If you, I see. Yeah. Yeah, good question. Yeah, that's right. So I did not show that, but we do have evidence that if I think this like in the embryo, if I make the embryo big, the concentration goes down, this structure doesn't assemble. It does have an impact on actually the rate of transcription. So it's a little chicken and egg. Transcription promotes condensation, but the condensation seems to speed up the transcription in those reactions. So I think that's reasonable evidence for functional. Yeah, that's right. But there are a few examples, clear examples, of the functional role of these things, and yeah. I mean, the devil's advocate is the cell's trying to avoid this because these liquids can be catalyze fiber formation and these diseases. I don't think that. The way I think about it is that these liquids are playing functional roles like regular organelles. The cell does this because it's useful, but the cell has to be careful because all hell can break loose, especially when you get mutations.
See, it's not so much chicken and egg, but rather it's Alzheimer's reciprocity. One promotes the other. Yeah. That's right. Maybe chicken egg is right, not the right term. Yeah, I think that's right. Yeah.
Why do you call those structures liquid droplets? They look like just lumps of sticky proteins. What's different from small pieces of actins sticking together?
Well, they really are liquids. I mean, I showed you the microbiology data where you can on these time scales and so forth, but they're liquids. They're liquids on those times. Yeah, well, the pathological things are solid, so that's not really true. But I think it's fair. Your point is fair that if you look at actin, maybe this is your point, which is totally valid. If you look at actin networks, long enough time scales, you're going to see a liquid. But which is why we have tried hard to access short time scales and see really when, you'll see signatures of elastic storage in water, right? If you access super fast time scales, so there's going to be, but on second time scales, which I would argue is a pretty fast time scale, these things are liquids. Yeah.